Welcome. Today, there is a YouTube video that we're going to start with as kind of a review of Section 4 guided notes, talking about how molecules move in and out. It's Amoeba Sisters. If you're watching on the video, you might want to skip watching it on the video. It's linked up above in the in-person agenda. So this does a nice job of kind of recapping how things move in and out of the cell, like Section 4 guided notes will. So we'll play this about 8 minutes long. We'll talk about a few things. Today we'll have some time built in for you to spend some time like reviewing, studying, getting some questions answered. So this is about 8 minutes long. There's a concept map, flow chart, graphic organizer, I don't know what you want to call it. This thing has a word bank, and some of those you might be able to pull from this Amoeba Sisters video that as you're watching it.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates, and when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside, and this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf, and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit, they sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. All cells have membranes, but not all cells have a wall, but plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so let's talk a few things, kind of general, kind of canvas things, before I give you a little bit of time here. So if we take a peek at the module for this week, so we're here today. This sheet right here is due midnight tomorrow. It's the flowchart thing that you have in front of you. Okay, there's a submission. The packet that you picked up in the back is called a study guide for cells. It's also due on Wednesday night at midnight before your test on Thursday. And if we take a look at this assignment, again, there's a Google Doc if you want to do it on Google Docs. There's a PDF if you want to do a PDF. If you want to take a picture of it, that's all up to you. But we also posted an answer key for you to check your answers after you've completed the study guide. So that the purpose of giving you the answer is just twofold. One, so you can check it to make sure you're doing it right, or if you say, I have no idea what they're trying to ask me to do, oh, that's what they meant for that one. Okay, that's the intent of that study guide, or answer key, I'm sorry. Don't turn in my answer key, because I already know what my answer key looks like. So there's that. Also, if you take a look on tomorrow's virtual day, there is this cells unit formative assessment. It's a practice to learn piece, it's an online Canvas quiz that I'm allowing you multiple attempts, and I'm gonna keep the highest score. It will keep the highest score for you. And one of the things that I'm asking you to do is if you do not get at least a C, then to take it again. And you can say, how do I know if I got a C? Well, it's out of 4 points. We're on a 4-point scale, so 2 out of 4 is a C. Okay? You are more than welcome to take it until you get an A. That's up to you, but I'm asking you to get at least a C on it. And it will be good preparation for your test on Wednesday or Thursday. This is an older version of the test. It's not identical to what you're going to be given. There are a lot of similar questions. You know, I can only ask so many ways, like what the function of different cell organelles are. I can only ask in so many ways what the diffusion of water is called. Okay, so there's that. Okay, so these are kind of some things that we designed to kind of help you prepare for your test. I want to talk through a few more things before I give you some dedicated time for your flow chart or concept map or graphic organizer, whatever you want to call it. I do want to discuss grades. So you are testing on the 4th or the 5th, and I am saying that if you have submissions in by Friday at 8 a.m., I will guarantee that they get put on your quarter grade. If you turn it in at noon on Friday, it may or may not make it. Okay, I don't know. I'm not sure when we're actually going to get to, I'm going to get to posting quarter grades, but realize a quarter grade is how are we doing right now? And that's based upon evidence that I have in the grade book. Now, with further evidence, my conclusions might change. But at some point, I have to make that decision. So the idea is, if you have it in by 8 a.m., I guarantee you'll be in there. I might not do grades till Monday at noon. I don't know when I'm going to get to them. So I may still be grading stuff as it comes in after 8 a.m. on Friday, but I'm not guaranteeing that I'm going to have that in your quarter grade. Even if I don't get it in on your quarter grade, it will still get graded. You will still receive feedback. It'll still be part of your overall semester grade. It just might not make it on your quarter grade, which is a progress report grade of how you're doing so far right now. Your semester grade is what counts for your transcript. Okay, so something to keep in mind there. On your osmosis lab, as I was grading the osmosis labs, there were some questions on the back about your potatoes that were not great. So I want to make sure we kind of address a few things here. When we talk about the water, there was a series of questions. And the intent was to try to walk you to the point of kind of determining whether it was hypertonic, hypotonic, or isotonic. So if we have our water, we have our potato soaking in some type of sugar solution. We purposely picked 0% and 25% because we're at the far ends. Water was either flowing in or flowing out. We could almost guarantee at that point. So let's just assume that we were talking about mass was gained. Now, because mass was being gained, what we're saying is water must have been flowing into the cell. That's where that mass came from. Now we kind of got to go back a little bit and we say, well, if water was going into the cell, then what must the concentration of the water been outside the beaker? So we got to go through and say, oh, osmosis, oh, it's diffusion of water, it says over there. And diffusion is passive transport, and it flows from high concentration to low concentration. So if water was here and it flowed in, it must have been high concentration outside to low concentration inside. So the concentration of water in the beaker was high compared to the potato, which is great. But what we really care about in terms of hypertonic, hypotonic, and isotonic is really about the solute or the sugar solution. So with these cups, I kind of kept referencing these cups. If this has a lot of sugar and this has a little sugar, which one physically has more space for water? The one with a little sugar or a lot of sugar? Cup 1 or cup 2? Which one has more water in it? Number 1, right? They're inverse of each other. They're always inverse. So if my water concentration was high outside, that must have meant my sugar concentration was low. And if my water concentration was low on the inside, that must have made my sugar concentration high. So that means the sugar concentration in the beaker was low compared to the potato. And then when we reference hyper, hypo, and iso, what we look at is the sugar or the solute concentration, and based upon the fact that it's low, it's hypotonic. Or you could say, hey, water is flowing in, it's kind of like an orange full of water, very juice, very plump, hypo, like an orange. Okay? And these pictures that we have on the board, go like that for the video, should be helpful kind of to, oops, I need the key, to kind of answer some of those questions. So hyper, hypo, isotonic, we're talking about the solutes. Does it get bigger and juicy and plump and increase in size like an orange, or does it shrivel up and decrease in size like a raisin? Hyper, like a raisin, hypo, like an orange. Okay, so I'm going to give you a couple minutes. In those couple minutes, I want you to go through and I want you to work on this graphic organizer concept map. There's a word bank. Each word is used once and only once. If you get done with that before we go through it, feel free to flip it over on the back. These pictures might be helpful for the pictures on the back. Okay, if you finish all of this, then there's a study guide packet that you can work on. If you finish that or you want to work on something else, you can work on the formative test. I did have a student who couldn't view an image on the formative test, and it ended up that they had updated their iPad to iOS 14, but they didn't go into their settings and toggle on that thing that they talked about to allow for, I think it was called cross traffic with the student app. So if that's the issue, make sure you go into your settings, find Student Canvas, and toggle on allow cross traffic, website cross traffic, something along those lines, and I can help you with that. So I'm going to give you a couple minutes to work on the concept map. We'll go through it, and then I'll give you some more dedicated work time to kind of prepare for the test and some of those other things. All right, let's talk about this concept map. So when we talk about how things get into and out of the cell, there's lots of different ways that we can kind of look at this. So there's two general kind of categories that we can break these things into. They either require energy or they don't require energy. So there are some things that we kind of try to use as kind of memory devices. If you eat a lot of food, that helps you become more, well, that's actually a bad prompt. Okay, let's start over with that. You're either active or you're very passive. Okay, those are kind of our two general categories. So which one requires a lot of energy? If you're active, does that require a lot of energy? Or if you're passive, does that take a lot of energy? Active, right? So when we talk about requiring energy, we talk about something called active transport. And if it doesn't require energy, we just kind of generally refer to that as passive transport. Passive transport. So let's spend a little bit of time talking. We're going to kind of focus on the passive transport. So passive transport was the example of you guys standing outside, mosh pitting, waiting excitedly to get into biology class. So when you're out there, you're like, whoa, biology, can't wait for biology. I know, I feel the same way every day too. So the idea is, when I open that door, it doesn't take any energy for you to fall in. Okay? When we talk about movement of molecules, we can say they either are moving from high concentration to low concentration or from low concentration to high concentration. In the example of the mosh pit, you're outside, I'm inside. Are you guys flowing from high to low, or am I flowing from low to high? Which one is happening in a mosh pit situation with no energy being required? From high to low, absolutely. Okay, so that's high to low is passive transport, no energy. Just kind of happens because of the constant random motion of your mosh pit. Now, when we talk about passive transport, it can either go straight through the phospholipid bilayer, like oxygen, carbon dioxide, water, or sometimes they need a protein channel. They need a little help. Transport protein. They need a little help. So if it needs a little bit of help, let's start with, if no protein channel is needed, it's simply referred to as diffusion. Sometimes it's called simple diffusion. That wasn't your word bank choice. So our choice is diffusion. So what is diffusion? Diffusion is the movement of molecules from high concentration to low concentration with no energy needed and no protein needed as well. That's that's diffusion. If it needs a little help and it has to go through the protein channel, then we call it facilitated diffusion. So what's facilitated diffusion? It's a form of passive transport that goes from high concentration to low concentration. No energy is required, but this time we need either a transport protein or we sometimes call them a protein channel. Kind of one and the same. Okay, so we need a little help. It's still diffusion though. It's still high to low. It's still no energy required. Because water is so crucial to life, they give the diffusion of water its own special term, and that term is referred to as osmosis. So osmosis is the diffusion of water. And when we talk about the diffusion of water, that's where the terms hypertonic, hypotonic, and isotonic come in. And use the things like hypothermia. Boy, your child is you are feeling very hyper. Your energy is above normal. Hypothermia, your temperature is below what it should be. Use those kind of, those root words. I guess I have the root words. Use the prefixes to your advantage. So if you're very hyper, your energy is higher than it should or above average. So we call that a hypertonic solution. An isosceles triangle has two sides that are the same, so that's isotonic. If you have hypothermia, your body temperature is below what it should be, and that's hypotonic. All of these things, osmosis, diffusion, facilitated diffusion, are high to low. High to low. There's no energy required. Some need a protein, some don't need a protein. If we switch over to active transport requires energy. Now there are some form, we're not ready for that. Active transport. Okay, so if passive transport is from high energy or from high concentration to low concentration, active transport is the opposite. That is the movement of low concentration to high concentration. That's me trying to get into the hallway when you guys are mosh pitting. It takes a lot of energy for me to go against that flow. If we talk about a slide analogy, or we talk about a river, if it's easy to go down the concentration gradient, it's easy to go down the slide. It's hard to go up the slide. It takes a lot of energy. I just got to sit there to go down, but it takes energy for me to climb up. If I'm floating down the river in a tube, I just float down the river, no energy is required. But if I'm going to go upstream, it takes a lot of energy. I really got to paddle and I got to work. Same idea. Active transport against the river, against up the slide, against the concentration gradient from low to high concentration. Sometimes, sodium potassium pump referenced in the video. Sometimes they use a protein channel or transport protein to do this big movement of molecules from low concentration to high concentration. It's for nerve impulse. Okay, and they'll use a form of active transport in a protein channel. These two terms are not related to protein channels. This is just like engulfing stuff or spitting stuff out. Right? So this is how coronavirus, SARS-CoV-2, gets into your cell. That's how once it's duplicated, it gets out of the cell. And the idea is moving into endo or exiting, called exo. We can kind of use those ideas. So into the cell is endocytosis, exiting the cell is called exocytosis, and they're both kind of generically called bulk transport or bulk movement of molecules. They showed in the video, the amoeba sister, they showed like, hey, this is how amoebas eat. They have these things called pseudopods that engulf and pull in and it becomes part of the cell. Endo, into, exo, exit. Okay? So, we have some time. We have actually a fair amount of time for you to work on the back page of this. Okay, these pictures on the board will probably be helpful for you to work on the back side of the worksheet. There's a study guide. This study guide, the answers are posted, is due, can be worked on and completed. There's a formative test that you can work on and submit. That's what you can do in this remaining time today. If you have questions, I'm here. Please ask, please ask, please ask, please ask. Test is on Wednesday or Thursday.